Good morning, student. Today we will discuss about structure of prokaryotic cell. In previous lecture, we will learn about cell, then the term cell, which is discovered by Robert Hooke, and the Robert Brown, which gives the name nucleus, the structure which is present in the cytoplasm of the cell in prokaryotic as well as in eukaryotic cell. But in prokaryotic cell, nucleus is known as the nucleoid because nuclear membrane, which is not bounded to a genetic material. Hence, in prokaryotic cell, genetic material which is known as the nucleoid. Then, in previous lecture, we will also study cytology, then theory, cell theory, modern cell theory, then totipotency. Then in today's lecture, we will discuss about the structure of prokaryotic cell in living organisms. In living organisms, the cells are grouped into two main categories. The cells are grouped into two main categories. First category is the prokaryotes and second is the eukaryotes. First is the prokaryotes and second is the eukaryotes. The prokaryotic term is also derived from the Greek word. That is the pro means primitive and karyons means nucleus, means the primitive nucleus which is present in the cell. These cells are known as the prokaryotic cell. In this cell, nuclear material is not bounded by a nuclear membrane. In these cells, nuclear material is not bounded, or genetic material which is not bounded by a nuclear membrane. Organisms like bacteria and cyanobacteria. Organisms like bacteria and cyanobacteria show prokaryotic organization. Organisms like bacteria and cyanobacteria Cyanobacteria, blue-green algae, which show prokaryotic organization. Cyanobacteria are the photosynthetic bacteria. Cyanobacteria are the photosynthetic bacteria. Are the photosynthetic bacteria. They may be filamentous or non-filamentous. They may be filamentous or non-filamentous. Filamentous or non-filamentous. Then many cyanobacteria which fix the atmospheric nitrogen. Many cyanobacteria which fix the atmospheric nitrogen. The motile bacteria possess a fine thread-like appendages called flagella. Which help in locomotion. Then pili help in process of conjugation. Then fabry help in clinging to a support. Then the next point is the structure of prokaryotic cell. A common bacteria, Bacillus, show a basic two layers, that is the plasma membrane and cell wall. The common bacterial cell Bacillus shows a basic two layers, that is the plasma membrane and cell wall. But some bacteria shows a three basic layers. Common bacterial cell which is oval in shape, which shows two basic layers. That is the plasma membrane or cell membrane, and next is the cell wall. Which is composed of peptidoglycan, which receives stain. Cell wall composed of peptidoglycan. Then innermost layer is the plasma membrane or cell membrane. But some cells show three basic layers, but some bacterial cell shows three basic layers: inner plasma membrane, middle cell wall, and outermost layer is composed of gelatinous material, hence called slime layer. Or capsule. The outermost layer, which is known as the Glycocalyx, This Glycocalyx layer which perform the function to prevent drying of cell or to prevent desiccation of cell. Outer layer, middle layer, then innermost layer. Then first Glycocalyx layer, which is the outermost layer of bacterial cell, which is made up of macromolecules. made up of macromolecules which help in adhesion of bacteria, Some bacteria have loose sheath called slime layer. Other may have a thick and tough covering. Called capsule. The Glycocalyx the gelatinous material which is present in this layer. The layer is called as the slime layer, which is composed of or made up of a polysaccharides and may contain proteins. The next layer is the cell wall. Next layer is the cell wall. It is the middle layer. It is present below the glycocalyx. Cell wall which is present below the glycocalyx. It provides definite shape and which gives support to the cell. Then chemically the cell wall is made up of peptidoglycan. And murine. When cell wall which is made up of peptidoglycan, the peptidoglycan which receives stain, and the cell which is called as the gram-positive bacterial cell. Which receives stain. Hence, the bacteria is known as the gram-positive bacteria, and murine, the cell wall which is composed of murine means the slime layer do not allow the stain to the cell wall. Hence, the slime layer or capsule which washed out the stain. Hence, these bacterial cells do not stay. Hence, these bacterial cells are called gram-negative. Gram-negative bacterial cells. The term gram, the Danish bacteriologist Hans Christian Gram, which developed staining method. Hence, the bacterial cells are the gram-positive as well as gram-negative bacterial cells. Then, next is the third basic layer is the plasma membrane. Third is the plasma membrane, or which is called as the cell membrane. It is the innermost covering of the envelope. It is the innermost covering of the cell envelope. Chemically composed of lipids and proteins. Chemically composed of lipids and proteins. Cell membrane is a phospholipid bilayer. Phospholipid bilayer. Then the cell membrane shows infoldings. The cell membrane shows infoldings. Which is called as the mesosomes. Mesosomes. Mesosomes which perform the respiration like a mitochondria which is present in the eukaryotic cell. Like this type of function mesosomes perform, means the breakdown of energy. Then cell wall, then which help in mesosomes, which help in formation of, which help in mesosomes, which helps in cell wall formation and DNA replication. Cell wall formation and DNA replication. when the genetic material means DNA which is attached to this site which is close to cell membrane or plasma membrane which attach to mesosomes, the DNA replicates and the half replicated DNA which is attached to this infolding and the cell divide by a conjugation process. Then some bacteria especially photosynthetic cyanobacteria show more longer extensions. Cyanobacteria shows more longer extensions called chromatophores. Chromatophores is the long extension of photosynthetic bacteria, the cyanobacteria. They contain different types of pigments. They are bacteriophlorophylls, bacteriophyophyte, and carotenoid. These three pigments. Then next is the cytoplasm. Next is the cytoplasm. Cytoplasm is a semi-fluid ground substance. Cytoplasm is a semi-fluid ground substance or matrix present inner to cell membrane. It contains variety of inorganic and organic compounds for cellular activities. They contain inorganic as well as organic compound for cellular activities. Cytoplasm does not show cytoplasmic streaming and lack membrane bound organelles like mitochondria, endoplasmic reticulum, lysosomes, etc. Then third part is the ribosomes. Ribosomes is the dense particles. Ribosomes is the dense particle which is present in the cytoplasm. In prokaryotic cell, 70S type of ribosomes are present. They are in individual, they form a chain which is known as the polyribosome. Ribosome help in protein synthesis. Ribosome help in protein synthesis. They are made up of RNA and protein. They are made up of RNA and protein. Then the prokaryotic cell shows 70S type of ribosome. 70S type of ribosome. S stands for a Sedgwick coefficient. Then next is the genetic material. Next point is the genetic material, which is not bounded by a nuclear membrane, which is called as the nucleoid, which is highly looped or coiled, double-stranded DNA. Double-stranded DNA, which associated with proteins, Then next point is the plasmid. Flagella, fembri, plasmid, plasmids are extra chromosomal self-replicating. Plasmid is a extra chromosomal self-replicating DNA molecule. In this plasmid, 3 types of genes are present. 3 types of genes. Three types of genes are present. On the basis of the presence of gene in plasmid, they are of 3 types. First type is the F type. Which is function as resistance against antibiotics. Then second type is the F type. F type. Second is the R type. Third is the col G. First is the R type or R gene. Second F gene or F type. And third is the col G. F gene plasmid form. F gene plasmid for reproduction. F gene plasmid for reproduction. Then R genes plasmid for resistance against antibiotics. F genes for reproduction. Second R genes which is present in the plasmids, which is responsible for resistance against antibiotics. And third is the col gene resistance against colyces. Resistance against colyces. Resistance against colyces. These three types of genes which is present in plasmid. Plasmid is a extra chromosomal cell replicating DNA molecule. Plasmids present in cells. Plasmid present in cells. The cells are called as positive strain bacteria. Positive strain bacteria. And plasmid absent in the cell. Plasmid absent in the cell. The cell is called as negative strain bacteria. Negative strain bacteria. The positive strain bacteria, which is also called as the donor cell, while the negative strain bacteria, which is also called as the recipient cell. Donor cell like a male and the recipient like a female. The enzymes which are present in the cytoplasm. The enzymes enzymes which are present in the cytoplasm. The enzymes which perform the function as a respiration, which perform all the activities of the cell, This is about the structure of prokaryotic cell. Thank you.